The formation of the nervous system, known as neurulation, is a critical event in the development of all vertebrates. The resulting structure, the neural tube, controls the development of many other structures in the growing organism. Neurulation begins during the 3rd and 4th weeks of embryonic development, after gastrulation. During gastrulation, 3 germ layers are formed: the endoderm, the mesoderm, and the ectoderm. Neurulation, however, primarily involves the ectoderm, with signals from the mesoderm. That prompt it to start elongation. The portion of the ectoderm involved is called the neural plate. The neural plate arises from the ectoderm under the influence of a structure called the notochord. The notochord is a cartilaginous rod in the midline that signals the ectoderm to thicken and form the neural plate. After being specified, the neural plate begins a process of intricate folding that will eventually yield the neural tube in the first hallmark event of neurulation, primary neurulation. Additionally, part of the ectoderm will become the epidermis. And between the forming epidermis and central nervous system is a presumptive neural crest. The first hallmark event of neurulation is primary neurulation. This process starts with the neural plate, which folds into the neural tube, giving rise to the future brain and spinal cord. The cells of the neural plate constitute an epithelial sheet, and the whole plate will fold along the dorsal midline. To start the process, the cells that lay along the midline, called the medial hinge point, or MHP cells, become apically constricted by rearranging their cytoskeleton. This brings the MHP cells down into a newly formed neural groove and the distal cells up into neural folds. The bending continues until the neural folds meet at the midline. Then, expression of cadherin proteins will allow the folds to zip together, starting at the caudal end of the embryo. Eventually, the folds are joined completely, and the neural tube stands complete. However, it is still connected to the epidermal layer by the presumptive neural crest. During primary neurulation, the neural crest cells, or multipotent stem cells, migrate throughout the body, giving rise to various systems and structures. By the end of primary neurulation, the neural tube has formed from the neural plate, and the neural crest cells have migrated across the body. The neural tube gives rise to structures like the dentine of teeth, Schwann cells, and the adrenal medulla, while the neural crest cells give rise to structures like the brain, the spinal cord, and the motor neurons. Following primary neurulation is secondary neurulation. While the mechanism is largely unknown, secondary neurulation forms the caudal end of the spinal cord. This is made possible by the condensation of mesenchymal cells into the medullary cord. Once the condensation process is finished, the medullary cord undergoes cavitation to form a hollow space, and the secondary neural tube is complete. Next, the primary and secondary neural tubes begin fusing together, eventually yielding the early spinal cord. Now the process of nervous system development can proceed throughout the remainder of gestation. Thank you very much for watching.